Good morning, everybody, and I would like to first thank the organizers for the invitation. It is a tremendous opportunity to be here, and I also would like to thank all of you for being here with us. So today I'm going to talk about sequencing our ways towards understanding and managing invasive species. As it is all well appreciated, this continual destruction of natural barriers due to increased global trade and human travel generates this massive biotic exchange between previously isolated regions and generates what we call this global biotic homogenization. Many ecosystems are dominated by non-indigenous species, and often these species have invasive traits that allows them to spread quickly and to also change the dynamic of invaded ecosystems, change species interactions. So this is particularly prevalent for the aquatic ecosystems. The major ports of the world are massively interconnected, and this is mainly due to the shipping traffic. So these ports receive massive amounts of ballast water and fouling organisms, and they often represent the entry points for non-indigenous species, thus our interest in studying these particular environments. But for now, I'm going to step back a little bit to introduce the problem a little bit better. So here are examples of notorious aquatic invasive species, and they have this bad reputation, as my title suggests. And this is because they have strong ecological and economic impact. And we spend lots of resources trying to sometimes eradicate them, but like often just to slow down their secondary spread. However, we learn from these experiences that they are particularly resilient. Yeah, they represent nothing more than successful evolutionary stories, yeah, evolution in action, species that constantly adapt to a changing world. And certainly there are important lessons that we can learn from biological invasions about the ecological footprint of our own species. So Elton's book, The Ecology of Invasions by Animal and Plants, was perhaps the first firm warning about an imminent ecological crisis. Yeah? He described the world as a very explosive world in which we don't know when or where the next outburst will be, but we might hope to find ways of stopping it or at any rate damping down its force. So with incredibly wit, Elton recognized this high level of endemism, regional and continental endemism, the continual destruction of natural barriers. Few years later, and precisely half a century ago, the influential book on the genetics of colonizing species, edited by Stebbins and Baker, was the first to place biological invasions in an evolutionary context. So the authors explore the changes that these species undergo as they enter a new ecological space or a new geographical area. Now, 50 years after, the field of biological invasions is going through massive transition. And more we use these molecular tools to understand biological invasions, and often we use barcoding and metabarcoding tools. So we use these tools to look at the identity of aquatic invaders and ask the question, who are these invasive species? And although these might look like a trivial task, in many cases their taxonomic status remains unsettled. We use also these tools to look at the evolutionary and invasion history and understand their native ranges and the sources of introduction. We try to also understand how they travel around the world, their vectors of introduction or vectors of secondary spread. Do we deal with single or multiple introduction events? And we use also population genetics or population genomics approaches to look at their colonization dynamics across invaded areas, look for signature of bottlenecks, or look for other evolutionary events, such as hybridization with native taxa, inbreeding, selective sweeps. So ultimately, we are interested in causes and consequences of invasiveness. But from a more practical point of view, and more recently, we can embrace barcoding tools to look for the early detection and preventing measures of biological invasions. So to give you a little bit of a feeling of how the work goes, during the first phase of the Canadian Aquatic Invasive Species Network, our group was involved in studying several invasive ascidians that had a major ecological effects. They have a global distribution. So here are a few examples of species that are commonly found along the North American coast. The colonial golden star, and violet tunicates, as well as the solitary vase tunicate. So these species can be found in huge aggregations on both natural and artificial substrates. So they spread rapidly, overgrowing almost all sessile species in this process. Now, the reason for this massive proliferation and rapid expansion is not well understood. However, it is believed that eutrophication, or pollution, dredging of the substrate, overexploitation are possible causes. So one of the first species that we investigated was the golden star tunicate, Botrytis loseri, which is considered to be native to European coastal waters and reached global distribution during the last 2 decades. It has a very strong impact on aquaculture facilities due to its ability to blanket and suffocate entire mussel lines, as in this picture. So as I mentioned earlier, so Botrytis is known to have a native range in Europe. However, this is mainly an educated guess. All we know is that this species reached global distribution during the last 2 decades, is found on most continents and major coastal zones. So our former student, Dan Bock, looked at the invasion genetics of Botrytis loseri using both nuclear and mitochondrial markers and phylogenetic and population genetic approaches. So this is the phylogeny, mitochondrial-based phylogeny that Dan recovered, and this suggests that Botrytis loseri is actually a complex of cryptic species. While one of these species, found here in blue, is highly invasive and was identified on all continents and coastal zones that we looked at, these other closely related species represented here in red and green have a very narrow distribution. They are found only along the English Channel. Now, you might be wondering if this genetic break, nice genetic break, represents biological species or reproductively isolated units, or they simply reflect genetic breaks due to historical barriers to gene flow, historical barriers. So when we're asking such questions, nuclear data can be very handy. So in this case, nuclear data based on microsatellite markers fully supports this mitochondrial phylogeny. So this is a Bayesian structure analysis with full support for 3 main groups that fully correspond to the mitochondrial clades. So in this diagram, each fine line represents a multilocus genotype, and the color code corresponds to the probability of that multilocus genotype to belong to its group, one of the 3 identified groups. And this is a very simple diagram because it reflects that most multilocus genotypes show almost 100% probability of belonging to their own group. And this was true also for populations in which these closely related species come in symmetry, like in this diagram. So you see members of the two clades here coming in symmetry. There is no evidence of nuclear gene exchange, suggesting reproductive isolation. So clearly these clades represent biological species. And this is important because such data sets allows us to explore the extent to which distributional ranges are phylogenetically constrained. Do closely related species show similar distributional ranges? And the answer appears to be no, at least for invasive species. We recover similar pattern in other invasive ascidians. So one question that these findings raise is, why do some species proliferate at an alarming rate while their closest relatives are facing extinction? Is this due to the competitive exclusion, one species being a better competitor than the other, due to the stochasticity of the invasion process, some species having access to vectors of dispersal and secondary spread? Or is it simply due to fine-scale adaptations that perhaps ecological genomics approaches can identify? But right now, I'm not going to go deeper into this question. However, I want to just suggest that these patterns of closely related species with different distributional ranges and different dispersal abilities pose a great problem when we try to identify invasive species. And I'm going to switch gently the gear now to look into the early detection of high-risk aquatic invaders in the Canadian ports, like both freshwater and marine ports. As most of you know and appreciate, these days it's very simple to combine the use of traditional barcoding approaches with next-generation sequencing to describe complex communities and to also identify invasive species. It's what we call metabarcoding. And with this procedure, we sample complex, so we get complex samples, as in our case, zooplankton samples. We grind entire samples and we amplify a particular marker of interest. In our case was the 18S marker. We obtain millions of sequences, we filter them, then we cluster them using different analytic clustering methods, and then optionally, when necessary, we blast representative sequences of these OTUs to describe these complex communities. So the method is very promising, however, has also problems that have to be worked out along the way. And for this reason, we try to validate our methods using a series of mock communities. So we assemble both very simple and more complex communities. For example, this is an example of a simple mock community in which each of the 20 species that entered in this community was represented by a single individual, generally non-brooding, and individually labeled. So this allowed us to look at the level of intra-individual genetic variation, if any. And then we assemble more complex communities in which each of the 30 species that enter into this community was represented by multiple populations, both population of small numbers and more abundant populations. And with this approach, we are able to look at the level of intra-population genetic diversity. So using this combination of mock communities, we are able to explore a number of questions. We look at first at the impact of intra and interspecific diversity to our ability to recover OTU numbers that correspond well to species richness. So we found that simple communities, the ones based on single individual per species, generated a generally high correspondence of about 60 to 84% between the OTU number and the species richness. However, when we went to more complex communities, when we examined them closely, we noticed that the correspondence dropped to about 30 and 63%. We investigated this further and we found that intraspecific and sometimes even intra-individual variation in our 18S region exceeded the 3% divergence. And you think that the solution is simple. We can simply adjust our threshold. However, we found that at the same time, many closely related species show less than 3% divergence. So we clearly had a problem that we had to overcome. Another element that aspect that we were able to investigate using our series of mock communities was looking at the effectiveness of different methods of clustering, different analytical procedures. So we use a combination of stringent and relaxed filtering with singleton sequences included and excluded. So if you don't remember, singleton sequences are those very, very rare sequences that you capture only once in your sample. We also use 3 different clustering algorithms, the ones that are commonly used in the literature, MOTHER, uClust, uParse, and also use different methods, 3 distinct methods of treating alignment gaps. So we investigated all these analytical procedures and we found that the number of OTUs varied by nearly 3 orders of magnitude when working with these different analytical approaches. We found that the use of relaxed filtering and the inclusion of singleton greatly inflated the number of OTUs. At the same time, the methods of treating alignment gaps had a strong impact on our estimate. So after going through these validation steps, we felt a little bit more confident in going and exploring the spatio-temporal zooplankton biodiversity trends across the 16 Canadian ports. So the Canadian Aquatic Invasive Species Network had a sampling team that managed to sample 16 ports across 4 geographic regions, Atlantic, Pacific, Great Lakes, and the Arctic. So 4 ports per region. Each port was also sampled at 4 different sites across 2 seasons and with 2 different plankton nets, different sizes. So it's a big sampling effort that was involved in this project. Now, in order to try to overcome the problems that I just mentioned that we encounter with this particular marker and with particular data set, we use 2 different methods of analyzing our data set. So we use the general clustering approach that I just mentioned, so to generate the OTU clusters. However, since this method requires quite a good, reliable barcoding gap and accurate divergent threshold, which we didn't have, we use another complementary method that we call a taxonomy-dependent approach. So in a simple way to put it down, this involves blast searches of individual reads against local databases. Yeah, so this method requires a well-developed reference library and certainly the concerted global effort. And it also depends on a local database to make it computationally feasible. Yeah, so it requires a customized pipeline specific to the marker and taxa of interest. And the taxonomic extension is at the core of this approach. So your resolution depends on the local database. The results that I'm going to report are based on this method because we found that it provided us much higher resolution and accuracy. So these are the major highlights, and I'm going to go into this point by point later on. But for now, I just want to mention that we analyzed 7 million sequences, and more than 90% of them were assigned, taxonomically assigned. However, only about 50% of these sequences were assigned at the species level. Regardless, we managed to identify species spanning 392 metazone families over 105 orders. And these families complemented previous survey based on morphological identification. We also revealed species from over 30 orders that were not previously reported that we are investigating now closer. We also found, as you would expect, that certain taxonomic groups were underrepresented to the incomplete reference database that we had to work with. And again, as you would expect, zooplankton communities were very distinct among coastlines and also among habitat types, marine, freshwater, estuarine. We found a little bit unexpectedly that biodiversity varied substantially across seasons, particularly for the subarctic region, with Churchill showing very high seasonality, not only in species composition, but also in abundance. And we also revealed a large number of non-indigenous species, with many of them in unreported regions. So 20% of the species that we looked for, we managed to identify. So now I'll go a little bit slower, step by step. So this is a proportional abundance of phyla that we identify across samples, like all the 16 ports. And you would see that, you can see here that most of the sequences, more than 90%, belong to crustaceans. And this was intended because our primers are intended to amplify crustaceans. We also amplify annelids, ctenophores, mollusks, and from time to time we amplify also groups that our marker were not intended to actually recover. If you go at the finer taxonomic level, at the family level, here you can see the proportional abundance of family across samples, with each column representing a port of the 4 regions that we investigated: Pacific, Arctic, Atlantic, and Great Lakes. So this diagram is a little bit difficult to digest, but the take-home message here is that the most abundant families that generally belong to copepods are also shared among ports within a particular geographic region. Now, a little bit more intuitive is to look at the PCA, principal component analysis of samples, based on the family level, based on presence, absence. And here you see the strong grouping. Each dot in this diagram represents a particular site or season in a port. So all the samples in the Great Lakes are here in the greenish colors, and then in red, the Arctic ports. Then with blue, Pacific, and purple, Atlantic. You can see that the bayside port form a clear cluster, very distinct cluster, because this was the only port that has a true estuarine environment. And for the exercise of identifying invasive species, we use our specific approach of blasting individual sequences in order against a local database that included 125 non-indigenous species. We identify 25 of these species, so we identify about 20% of the sequences that we entered in our database. Most of these non-indigenous species were recovered in the Atlantic ports. So this afternoon, our postdoc, Frederick Chain, is going to continue this story about our exercise of identifying invasive species. So if you are interested in our protocol, in the way we validated this methodology, please join his talk around 4 o'clock in the biosurveillance session. Now, before closing, I would like to tell you a few words about the things that we learn from these projects and from working with 18S sequences. So clearly the metabarcoding approach is very sensitive if applied appropriately. We still have major steps to do to go to further advance this methodology. We greatly need reference libraries that incorporate reliable taxonomic information. There is no question about this. We also need comprehensive databases of invasive species. We made a good start here, but there is a lot of work to be done. We also need methodological validations. More of these validations are needed using more communities to calibrate our estimates. We also, I think, need to make this transition to multi-marker approaches. And if you'd like to see the progress we are making towards this approach, I invite you to join the poster 15C or C15 that our master student Zhang is going to present on Wednesday. And of course, we need to continue to promote standardization and reproducibility of metabarcoding approaches. And all this can be only done to a global coordinated effort that integrates traditional approaches and effectively implements the emerging technologies. So I would like to send a big thank you to my long-term collaborators as well as to my enthusiastic lab members that always bring on board the unexpected element of the research. Special thanks to Julian, Emily, Freddie, Guang, Eben, so they ultimately run this research. And I would like to thank the funding agencies. Thank you all for your attention. But before I'm closing, I would like to extend my thank you to Paul Hibbert and his enthusiastic team at Bio for making barcoding a Canadian icon. And to close this picture, I would like to invite all of you to consider the NRC Canadian Journal Genome as a destination for your exciting research. Thank you very much.